Welcome to the webinar of the COMMeND Network for Otitis Media Researchers. Today we have the pleasure of two of our mentors to give the webinar talks. The first one is Dr. Joseph Kerschner, a pediatric otolaryngologist who became the Dean of the School of Medicine and Executive Vice President of the Medical College of Wisconsin in November 2011. He then became the inaugural Provost of MCW in 2017 and served in these roles until January of this year. Dr. Kerschner also maintains an active research laboratory, which has received extramural funding of more than $14 million. With that, he has published groundbreaking basic and clinical research in otitis media, including over 170 manuscripts in peer-reviewed journals and authored or co-authored 20 books, chapters, and reviews. He provides editorial expertise for the leading national and international journals in otolaryngology and is the editor-in-chief of the International Journal of Pediatric Otorhinolaryngology. Currently, Dr. Kerschner is a senior partner at Chartis Consulting, a senior scholar at the New York Academy of Medicine, and has co-founded two research startup companies based on his research successes over the decades. And today it is quite a different format than our previous webinars because we asked him to talk about two things, which is scientific writing and his research on middle ear. So he will just be giving one talk for these two topics. Thanks, Joe.
Thanks, Reggie. Thank you for that nice introduction, and thank you all for being here. I've been able to meet some of you and hope to have ability to meet all of you in the upcoming year. My email is now Kerschner Joseph. So if you see the last name and the first name, and then you put 100 after that at Gmail. So anybody that has questions, you can certainly reach me in that way. If anybody wants a copy of these slides, Reggie, you can work to make sure that they get them. I put the affiliations below. While I'm no longer with the Medical College of Wisconsin, I still have lots of close contacts there, still have the laboratory people there that I'm continuing to work with and mentor, and have a number of grants that I continue to be maintained on through these research startup companies that Dr. Santos Cortez mentioned. These are some of the grants that have supported this, and it's really been my honor to have received so much funding from your taxpayer dollars through the NIH for this laboratory. It's honestly been the greatest joy of my career. So I'm going to talk about two things, as Reggie said, and I did send some questions that sort of dovetail into these two talks. Hopefully, you all will have a chance to at least look at those and try and answer those. Reggie, I don't know when they answer them or if they answer them after the talk, but anyway, I like this slide because it starts as a fairy tale or a story. When you get to a certain point, I would say old, but when you get to a certain point in your research career, you can start this way once upon a time. So I think you all, as aspiring otitis media researchers, should aspire to some point in your career to be able to have a slide that says once upon a time. And there was the chinchilla, there is the chinchilla, and which I think many of you know, but it's very much like the human. It's a outbred animal. If you look at this homology with the human, actually we published this paper. Can't remember what the year was, but it says up here 2010. And actually the chinchilla DNA is more close to the human actually than the mouse. But when we started our laboratory, it was really almost impossible to do molecular studies, specifically using the chinchilla because it was so difficult to create the tools that you needed to do RT-PCR and those kinds of things because there wasn't a chinchilla genome. We didn't have those tools to be able to create the laboratory tools that we need. If you go and ask some of my laboratory people, PJ is still there, she will tell you how many hours and months and years she spent developing reagents for some of these molecular studies in the chinchilla. So I have a couple of these, knowing that the chinchilla is an outbred animal, though close to the human, you wouldn't have any good tools to use necessarily. So this will be a question that I ask to all of you. And I put this here intentionally because I think if you're a researcher, if you aspire to be a researcher, Certainly, crying is a natural thing, and that's all okay. But giving up and changing fields is always, I think, a thing that creeps into your brain when you have 10 grants in a row that are declined, or a paper that's refused, or your research data makes no sense to you or anybody else. And no matter how much you torture the data, you can't actually make it tell a story. So, but for me, the passion was always the children who suffer from otitis media. So that for me is the driving force. And I would say for those of you who aspire to research careers, find something that you care about, find something, an area that you care about. Obviously, otitis media is the most common reason for children to see a pediatrician in the United States. Huge numbers of antibiotics are given that, and it's also the most common and difficult reason for children to have hearing loss. So it is an important disease, and so I did cry a little bit. I didn't give up or change fields. So what did we do? One of the first things that we did, and collaborated with my colleagues at the University of Pittsburgh, and at Children's in Columbus. And it was Garth Ehrlich and Lauren Bakelitz, as you see on this paper, as well as some of the people from my lab, as well as some people at the Broad Institute. And we actually didn't give up. We just sequenced the chinchilla genome. And while that may seem like a crazy thing to do, and it was expensive and probably all in, cost us over $1 million to do it. We did sequence the chinchilla genome, and it has made the ability to do molecular studies in the chinchilla much, much easier. This genome is still housed at the Medical College of Wisconsin. It's on the RGD, which stands for the Rat Genome Database. Our medical school was the first to sequence some important rat models, and so we harvested that database, and it's there. And anybody that cares to, we can certainly point you into that direction. It is a wonderful tool that is usable. But what, so we have the chinchilla genome now, and we can do molecular studies in the chinchilla, but they're still difficult at times. They have small litters, they have a longer gestation, they're really expensive, and you can't manipulate the experimental conditions as easily or rapidly as you want. Obviously, they're not humans. So what would you do? Give up, cry, change fields, etc. And the answer, as you'll get from this talk, is no. One of the things that we were doing concurrently as we were struggling with the chinchilla is developing middle ear culture material to be able to use. And so back in the dark ages when dinosaurs roamed the earth, this paper from 2007, there was a single cell culture that had been developed a long time ago by David Lim at the House Ear Institute and his colleagues. It was a reasonable tool to use to help us accelerate some of the ideas that we had in the laboratory. There were some difficulties, though. It was created from a 50-year-old man with really no demographic information. We didn't know what this individual's ear status was. We suspect, because it was created at the House Ear Institute, that it was somebody that was either going for a cochlear implant or somebody that was having a acoustic neuroma that was removed. But we didn't know if they had ever had a history of ear disease, middle ear disease, I should say. And then this cell culture was relatively old. After many, many passages, it was losing some of its foundational characteristics. There were difficulties with contamination of the culture material, and there were lots of questions in the scientific community about what we should do. So here's my slide again. There really were times where you almost said in my laboratory, yeah, we're not going to do this, and we're going to change fields. But we didn't. We continued to shed a few tears and move forward in a number of different ways. And one of the things that we did, and this would take about a decade, and it's come to fruition now, is that we decided that as a laboratory, we would develop the new cell lines for the future. We wouldn't rely on this old cell line that had all the problems. And I'll talk to you a little bit more about that as we move forward. But Science does not stand still. And so, what could we do in the meantime? Well, we developed this technique of primary cell culture from children who have ear disease that I operated on as a surgeon. And good, bad, or in between, our laboratory was the only one that can consistently develop this repository of primary, not immortalized. And so we can talk a little bit more about the difference between the older cell culture I mentioned and the ones that we were developing. But these are primary cell cultures. So meaning we had to get the tissue from the children each time that we did experiments with these cell cultures. My team was very, very, very expert at doing this. And it is difficult, though. Most surgeons don't want to spend the time to collect the tissue, don't necessarily have the expertise to do it. But this did propel a number of experiments forward and allowed us to really understand what was really happening in the middle ear epithelium of children. Probably foundational to most of those grants that you saw on the first page was this work. And so we established these from patients 6 months to 12 years, but we importantly had a control population. So you'll see the second two bullets there, the recurrent otitis media patients. So those are patients that have recurrent middle ear disease, but don't necessarily have chronic middle ear effusion. And we were able to sample tissue from those children, as well as the kids that have OME. But we needed controls. And so we developed the tools to be able to harvest this tissue from pediatric cochlear implant patients. I can tell you that that's not easy to do. As a matter of fact, it's quite difficult and took us some time to be able to get there. But we had well-established demographics, and we had multiple considerations around sex, race, and other demographic kinds of things. We did need tools and standardized techniques. I would harvest. I was the surgeon on all of these cases, less than a millimeter of tissue from near the eustachian tube orifice, and then place this in the culture material. I've skipped a lot of the steps, but those are the basics. And it came forward with a very consistent ability to do this. We obviously had to have IRB approval for this. There were a lot of considerations about IRB approval, but as we developed these techniques and were able to show that the children did not have any difficulties from this technique, it made it easier for us to go back to the IRB for additional studies as we went forward. So the middle ear epithelial, without giving too many of the details, and there are many more here, but these are just the basics of how we maintain these cells and grew these cells, and then also went on to immortalization of these cells with SV40 antigens, and as well as some other techniques that I won't put in here. But so these are just some representative images of the middle ear cells in culture. You can see that we did lots of studies from immunofluorescence, looking at their cytokine ability, looking at other markers to show that they were actually consistent with middle ear epithelial cell cultures, and some of the other kinds of things that we used to compare these cell cultures as we move forward. We did do, as we went to the immortalization of the cells, we did do cytogenetics, and we're able to demonstrate similarity to non-immortalized cell lines, and that was an important step. And we ultimately published all of this work, and I think I have a few of those papers in here. But while we were doing all of that work, as I mentioned, we were progressing along our hypotheses around what really happens in otitis media in the middle ear space. And one of the things that we knew early on was that mucins and mucin mucoglycoproteins were really important. And so mucins in any epithelium is really, really critical for the protection of the epithelium to prevent invasion of pathogens. But the flip side to that is that mucins also are what cause the difficulties around chronic middle ear disease, as well as hearing loss with these children. But the first step was my triological thesis. Triological Society is a society within otolaryngology. The reason there's only one person on this paper that I published back in 2007 is back in, again, the dark ages. They only allowed one author on these papers, even though others contributed to it, and you could acknowledge them in the paper at the end. But we set out to really show what was happening from the middle ear gene expression, knowing that the mucin genes were important for this middle ear epithelium, and published a number of papers on this. What I guess I'll say with this is that we did compare this then also with the cell culture techniques that we had, and we're able to show, as you can see on the RT-PCR homology across both our primary cells that we got from the middle ear, as well as our cell cultures, which was important again to demonstrate the benefit or the utilization or the utility of our cell cultures. We showed that there were actually 19 human mucin genes, and that this was a much more complex picture than people had appreciated previously. But we really did emphasize the gel-forming mucins, which we knew would be primarily in difficulty with these children who have hearing loss and chronic otitis media with effusion. And I'll tell more about that story in a little bit. But also part of this was driving this concept that otitis media, when I came into the world of otitis media research, there was a lot of talk about the eustachian tube and how the eustachian tube maybe was important in creating middle ear disease and otitis media. But we really felt like that otitis media was primarily an inflammatory disease, one that had important aspects of host microbe interaction. There were important genetic aspects of it, and that it wasn't simply an anatomic issue. And so we were really bent on trying to uncover or unravel that aspect of otitis media. And one of the concepts that didn't come from my laboratory, it really came, as you can see on this paper here, the second to last and third to last authors, Dr. Post and Dr. Ehrlich, who were wonderful mentors for me and really helped with this work early on. But we really felt that otitis media was a biofilm disease. I won't go into all of the details because we don't have time this morning about biofilms and all of those things. But I'm old enough to tell you that when I went to medical school, we didn't even talk about biofilms. The concept that biofilms was a human pathologic paradigm didn't actually exist. Kind of weird to say that in one human's scientific career, that you can see this paradigm shift that happens. But biofilms were known to be important for engineers looking at bacteria in nature, and 99% of bacteria in nature lives in a biofilm environment, which is a low metabolic state where they have a glycocalyx that protects the biofilm from environmental attacks on their community and their ability to survive. And so we really thought that this was what otitis media was, is a biofilm mediated disease. And we published this paper in JAMA back in 2007. Probably will be the highest impact paper that I ever publish, because I do tend to publish on niche things in more niche journals. But this was in the actual JAMA that is published and people all over the world read. But it was really proof of concept. We had been able to show, my collaborators have been able to show, that biofilms were otitis media in animal models, chinchilla in particular. But this was the first in which we were able to show that biofilms really existed in the human. And what we were able to demonstrate using that model that we had developed about controls is that 92% of the patients who went to the operating room for chronic middle ear disease, so either recurrent otitis media or chronic otitis media with effusion, had biofilm mediated disease, whereas our control population, in the study design, I would never have anticipated it would have been this stark of a difference. But in the control population, which were our cochlear implant patients, and we were assiduous in finding those patients, they couldn't have had any more than one ear infection per calendar year of life. So we really took, I would say, naive patients from an otitis media world And none of the cochlear implant patients had any biofilms in their middle ear. So this was just really amazing. We developed all kinds of tools which are in this paper if you care to have something to put you to sleep at night. But I put some micrographs in here, and it was a great paper, and my collaborators were kind to put me as the senior and last author on this paper. Obviously, we did a lot of the work in my laboratory, but I was still pretty junior at the time, and this was really helpful. The lesson here learned is trying to coordinate these talks about publication. As you develop your scientific career, think about who is with you on a publication. Think about where their career is and how you might help them, how you might mentor them, how you might help them in designing a paper, but also in writing it and then bringing that forward to the scientific community. This for me was a landmark paper where the generous support of my mentors to include me in the senior position on this paper was foundational in my career moving forward. So just a nugget from that perspective. Um, yeah, so this is a busy slide with a number of different publications, and I won't bore you with all of the details of these publications as they went along using these techniques. But essentially, what we did was using both cell culture techniques, but also these primary cultures, we dug into the mucin genes and the response to inflammatory pathways, and most importantly, perhaps hearing loss as things went forward. We importantly in the bottom left looked at how we could modulate mucin gene expression in some of these models, and that'll be important as I talk to you about sort of our next gen experiments that are going on right now in the company that Reggie mentioned. But this was fun work for me, I'll say. I hope it's okay to say it was fun. We did all kinds of experiments with great publications, and the team was prolific in many of their publications and ability to create data sets that I think were meaningful and again help to unravel what was really going on during the inflammatory infectious processes within otitis media. Um, we did continue to use a variety of different techniques, both these cell cultures and in fact the animal models were important with all of this. But as you see in this paper on the right, we then went from sort of the native cytokines to actually looking at bacterial components within these cell cultures and really honing in on these three mucin genes, MUC5AC, MUC2, and MUC5B, and looking at differences between non-typable Haemophilus strains, looking at strep pneumo as well, and Moraxella catarrhalis, which, as I think you all know, are probably the three most important pathogens when it comes to otitis media. So again, a lot of fun work went into that, and we published and presented a lot. Oh, one thing I wanted to say is, I'm sorry, I'm going back, but that biofilm concept, I presented. So there's the meeting. Some of you have been, but there's the ISOM meeting, International Society for Otitis Media meeting that happens every other year. We just had one in Philly. Kevin is the president. Dr. Mason at Nationwide is the president, current president of that organization. We just had a board meeting. I think we're going to the Philippines, Kevin. Thumbs up if that's pretty close. Yep. So for that meeting in 2027, I hope some of you can come and can convince your labs to bring you. But what I wanted to say about that is that the biofilm concept, I presented that concept at an ISOM meeting in 2005. I pretty much almost got run off the stage by a lot of people. They're like, "Oh, what's the data?" And I mean, it's a nice enough meeting. People weren't totally mean, but it was interesting. And within the 10 years, so by the time we got to I guess it would have been I don't know, my math is getting fuzzy. Yeah, I guess it's every 2 years. So it would have been the 2015 meeting. By the, like when I looked at the 2015 meeting, like the biofilm concept of otitis media was referenced in like something like 70% of the presentations or something, or maybe it was 65%. My point is, even in a decade with good science and good publications, the second nugget about publications, you gotta publish your work, you gotta publish it, and you gotta make sure that people know what you're doing. We were able to turn the tide on sort of people's thinking on that. All right, so we learned a lot, but we needed to get to the bedside. I'll say it that way. I think it's an important piece of research. Certainly, as you write grants, you all will be mentored into the okay. What is the what? What is the what for the patient? Right. Ultimately, science in medicine, yes, there's plenty of great work, and you can see from the papers I've published, a lot of it was basic research, and I don't know, maybe it had something to do with knowing the 19 mucin genes. I don't know. I mean, maybe that was important, but what is the what for human disease, and that's not necessarily the case if you're in the physical sciences. I mean, yes, there is oftentimes a connection to humans, but not always. But in the health sciences, the connection to humans is important. And so, honestly, from the very early days when I was battling Tyrannosaurus Rex and the dinosaurs, I mean, I always came with that concept of what is the human component of this, and I think the third nugget about publishing is. As you think about publishing your research findings, make sure that you have that upfront and center and at the end. Think about it both internally in your own brain, but think about it in your publications and make sure that it's clear. Even if you're publishing more in basic research types of journals, make sure that that connection is clear and weave that story or tell that story across the years, and in my case, across the decades of where you're going with all this. I oftentimes concluded my papers with next steps and where we're going. So anyway, moving to the human and the bedside, we wanted to know from all this mucin work about what were the most important mucin genes, and finally we sorted out from these 19 down to these two, and it's pretty clear that MUC5B and MUC5C are the two most important. They're gel forming, and maybe it made sense, but you needed the data to show it. And so we actually went forward to do this study, which I think was absolutely landmark and also a JAMA paper, where we looked honestly at the biopsy specimens that we were doing from children and correlated them to their overall hearing loss. And what this paper actually showed was as you showed greater upregulation of the MUC5B gene, you actually correlated that to more difficulties with hearing loss in these children. So it was the first paper that ever said, okay, we know that these kids that have bad hearing loss from their otitis media, they have this MUC5B gene that is correlated to their hearing loss. And so it really formulated a pathway to what are you going to do about it? If you know that that's the case, what would you do about it? What could you do about it? Can you regulate the MUC5B gene? Can you do something to really make this pathway better? And the answer we hope is yes, but it did create this set of questions. I will not answer all of these questions in my research career. I'm 60 now, and I'm catching up to where the dinosaurs all went. But I'm hoping that some of you on this call and your colleagues will find the path to doing this. But as a physician, as a surgeon scientist, the question would be, can we identify a subset of patients with mucin gene expression who have poor outcomes? Can you tailor that to personalized medicine? I fundamentally believe that the answer to that is yes. And that as a surgeon, when you go in to put a set of ear tubes in, most of these kids need ear tubes. When you put ear tubes in, could you do a sampling of their middle ear mucosa and identify who these patients are who are going to do worse? And how would that change your management of them? Would you Would you treat them medically differently? Would you treat them surgically differently? Would you use a different kind of tube on these patients, a longer acting tube? All of these kinds of things. Would you follow them more closely in your clinic so that when their tubes come out, you can check their hearing more readily? I didn't give you this paper. One of the fun things that we showed um, is that there's tandem repeats in these mucin genes. So while I talk about MUC5B as a sort of this construct, um, many people have different MUC5B expression, right? So they have these tandem repeats in the middle, and these genes make a mucoglycoprotein that's long, and these tandem repeats have an impact on making them stickier and more problematic. And so literally there's a whole pathway of research that could be done. Some might point to the fact that I spent 14 years in the dean's office, slowed me down on getting to the answers to some of these questions. There's probably some truth to that. But again, I won't go through all these bullet points in excruciating detail, but some of these things we have done. We've created cell culture banks from otitis media patients. Those are all available. They're commercially available. We couldn't do it in our own lab on our own, so we literally gave the material away to a commercial company, and they sell them. They're too expensive. I don't know what to tell you. They're maintaining them. I think it's $5,000-6,000 for a single aliquot of some of these cells. But if you have an NIH grant, you can certainly afford that. It can be hard for labs that are starting out. We have given away some of these cells. We've maintained some in the laboratory. We have given some to startup laboratories that have asked for them. And so, but we've made this available to the scientific community at large. Long-term outcome-based studies, we haven't done. New testing methodologies for mucin gene expression, we haven't done. Actually, I think that's relatively easy to do, especially as that technology moves forward. In other words, when Joe Kerschner says, could you tell something about a patient when you're in the operating room? If it takes you 2 weeks or longer to know what that patient fits into, that won't work. And so the rapid increase in technology can help with this area. The one area that I probably spent so it's the first and last bullet point that my lab has spent the most time in, and this transtympanic solutions is really where I'm spending the most time now. And I'm going to tell you or talk to you a little bit about that as well. So the tympanic membrane is it's an obstacle in the treatment of otitis media, treatment of biofilms. So it's skin, right? So it has tight junctions, right? So some of you may be aware of all of the different ways in which people are treating patients, disrupting those tight junctions, and delivering biologics through skin. So essentially, what it is you put a patch on somebody's skin, and it punctures those tight junctions, and then you can deliver your biologic through the skin. And there's a whole host of different entities where that works. The problem is the eardrum is down a centimeter or two from the opening of the ear canal. The patients you're trying to treat are children, and so that technique is not very effective for kids. So what you have to do is disrupt that tight junction in some way, or you have to get your biologics through that tight junction in some way. All right, full disclosure. I think it's important in science to be honest. So nugget number 4 about your publications, be honest. I mean, I'm the editor-in-chief, as Reggie said, of this journal, and I meet with lots of editors-in-chief of scientific journals. We're in a time where it's increasingly easy to be dishonest in science. There are ways in which you can manipulate micrographs. There's ways in which you can manipulate your photos. There's ways in which you can use other people's work. There's ways in which you can use chatbots to scrub research databases to harvest other individuals' ideas and count them as your own. I'm going to tell a quick story. I'm a storyteller, sorry. Reggie told me I had till close to the top of the hour. I promise Reggie I'll be done on time though. Um, but um, but I just had an instance at the Medical College of Wisconsin where 7 years of our work in this space, there was an individual who tried to harvest this data and claim that it was their own. Now, we've been able to refute that. We have 2 patents on this work and all of those other things. But I only tell that story not to say that there are demons in the world, but to tell you that there are people that will try and take shortcuts, and there are no shortcuts in science. There are none. Yes, you can get better, and I'm a big believer in AI, and you should use AI for its good purposes, not its bad purposes. But there are no shortcuts in science, and so as you get to a point where you're publishing your material, make sure that you are honest and do things in a good way. As the editor in chief, I can tell you there are many, many papers that come into my journal all the time that are clearly written with AI and not done very well, and doesn't seem like the articles are original. There are people that are taking different kinds of data, and they're creating these publications that are have a lot of falsehood to them. So I don't think I probably have to tell this group. You wouldn't be in this cohort if you weren't upstanding humans, but there's a temptation there because publish or perish, get the grants, et cetera. There's a temptation um, to um, to to fall down on this. So, all right, enough of the sermon. Me being honest is this last sentence, this issue will be solved in the next 5 years with biomedical engineering, the tight junctions. Full disclosure, I think I've been saying that now for 3 years. So if you're keeping score at home, that means my company has 24 months to get this done. I'm not sure, full disclosure, that we're going to get there. So I'm going to keep saying 5 years because then that just pushes it out. But for this small little group, you know that Kerschner's been saying this for a while already. And so he doesn't mean 5 years from today. Um, so here's the maybe the most fun thing that I've done in my scientific career. So with my cohort in our laboratories, Amit Joshi, genius person, biomedical engineer. He and I have been working on this for 7 or 8 years. And so what we've discovered is that if you take nanoparticles, liposomes, they can carry all kinds of cargoes. And actually, if you charge these nanoparticles, you can actually drive them through an intact tympanic membrane. And so we are able to deliver, at least through the chinchilla tympanic membrane, high concentrations of nanoparticles into the middle ear space. And they carry the cargoes of the things that we care about: antibiotics, steroids. And so if you were paying attention and you saw that paper where I talked about hydrocortisone regulating mucin genes, we've already done the work to know that you can regulate these mucin genes, which we've already shown are problematic in hearing loss in children. And we know that these biofilms, the reason that we have difficulty in eradicating them from the middle ear space is because we can't deliver a high enough concentration of antibiotics to the middle ear space by oral antibiotics. So they go through the mouth and through the GI tract, and they cause all kinds of complications. And they can't get to the middle ear space in a high enough concentration. And so they're not effective. And so the biofilms develop resistance genes, et cetera, et cetera, et cetera. So if you study otitis media, all of those problems are problems that we faced and that we know because as a surgeon, when I put an ear tube into a child's ear, the only thing we ever do to treat those kids' ear infections if they have problems after those tubes are in is to put a drop in their ear that goes right through the tube and into the middle ear space and cures their infections, cures their draining ear in 99% of the cases, cures their hearing loss, et cetera, et cetera. So we have a paradigm, but you can't put an ear tube in every kid that has ear infections. Millions and millions and millions and millions of kids get ear infections every year. Can't put a tube in. You can't put a hole in their eardrum. So you need this new paradigm of how you might be able to deliver antibiotics across an eardrum. And we have solved it. There's 2 other laboratories in the world who have also solved it using different things. Um, my company, I presented this to some venture capitalists not too long ago, and I said to them, I actually don't really care whether we make money on this or not, my goal is to help children, and I hope our technique works, but somebody's will. And the venture capitalists said they were offended, and they didn't ever want to hear me talk like that again. So yes, they have shareholders and they want to make money, but this idea I think definitely can work. We've got an NIH grant going in around this. Our phase 1 clinical trials will start in 18 months, which is why I'm telling you that I don't think I'll have it solved in 2 years. Maybe it'll take us 24 months to get to the phase 1 clinical trials. And then from there, if we show efficacy in phase 1, then it's off to the races. Then it really is off to the races with funders coming in, giving the ideas to somebody else who will take it across that path and go forward. But I hope you've been able to see that this is all woven together. The story that I've told from the days of the dinosaurs is all woven together around a human patient. I don't know that I ever thought I would get to this point in my scientific career to actually have a biologic that likely could really fundamentally change the way in which we think about ear disease and ear infections. But here we are, and we're on the cusp of it. And whether or not the Kerschner Joshi model works, or whether it's one of the other laboratories or somebody who comes after us that pushes this envelope forward, this envelope is going to be opened, and it will fundamentally change everything we know about otitis media and how we treat it and the research that comes next. So I'm telling you here that this envelope is going to get opened at some point, and it'll be an exciting time for researchers because it'll deliver a whole new paradigm of things that can be studied. All right, enough there. Just a few more nuggets on scientific writing from the position of somebody who's been a faculty member, a mentor, and a journal editor. But and I apologize if some of this sounds preachy. It's meant to be. If you were in my lab, you would get a little bit of this over time. But I don't think we spend enough time writing. So it's hard work. But surround yourself in your laboratories with people who like to write, who are willing to write with you, who are willing to critique your work, but who are also more than anything willing to help you. I just worked on a paper for a medical student. I actually won't ever be seen on this paper, but one of the faculty who was helping to mentor this medical student came to me and said, "Look, we're not making progress." And the senior mentor for this medical student is a busy surgeon, and he just didn't spend the time with the student and the junior faculty member writing the paper, didn't spend the time saying, okay, this is what needs to happen. This is what could happen. And this is how we should be thinking about this in a different way. And so the paper didn't move forward, and it was dying on the vine, so to speak. And so I think people want to publish. I mean, I don't think anybody does a research project or is in the laboratory or whatever that says, "Oh, I'll never publish this." But it often doesn't happen. This paper was really interesting. It was looking at sort of the factors influencing successful peer-reviewed publications, and my group published it, but many other people contributed to it. But this was from, if you don't know, ASPO, the American Society of Pediatric Otolaryngology, is arguably the most influential pediatric ENT society in the world. Big meetings, lots of people come, great presentations. But this paper was stunning to me. So some of the best research in pediatric otolaryngology, some of the best considerations in pediatric otolaryngology are presented at this meeting, and 35% of the people from this meeting never even submitted their work. Hard to get something published if you don't submit it. I mean, you can maybe paint it on a rock and put it outside on your driveway or something, and maybe somebody will see it. But this just floored me, and And the number one reason was time constraints. So people cited time constraints, like I did the work, but I just don't have time to publish. So I'm going to give you number five nugget. Um, a lot of people hate this nugget, but I'm going to give it to you anyway. I don't care. Um, Try and write your paper before you present it at the meeting. I don't mean like finish it, but try and write your paper before you present it at the meeting. Um, One of the things that came out of this publication in 2015 is now ASPO. If you're going to win an award, one of the awards that they give for your research presentation at ASPO, your publication has to be turned in before the meeting. So they're not going to make a determination on the award based on your abstract or what institution you're from or that they like the idea, but you actually have to have published, or sorry, submitted your paper to the program committee. I think that's really smart because once you get to that point where you've got it, I mean, it's not finished, right? Maybe, but it's good enough that somebody could read it. I think you'll publish it. All right. I think I have two more slides, and the last is just an acknowledgement one. But just to tell you that the publication process is more random than you might think. There's biases of the editor in chief. I have my biases, my associate editors do, the editorial board, the reviewers. There's time pressures on all the people in the process. So just like there's time pressures on publishing, sometimes people don't always give their best review. And there's just an overwhelming amount of material. If you don't know, the publishing of scientific papers, by and large, is a volunteer business. As an editor in chief, I get a small stipend. My associate editors, I have 10 of them that run the journal, get a very, very small stipend. I mean, basically nothing. And the rest of the people who are on the editorial board and reviewers, they're volunteering their time for the good of science. So why do I tell you that it's far more random? The reason is don't get discouraged. I do believe if you believe in your science and your data set, even if it's rejected, take the comments that come back from the reviewers and try again. Think about where else could I publish this? Should I publish this? Did I get a comment that says, this research is fundamentally flawed because of X, Y, and Z? Okay, if it's fundamentally flawed because of X, Y, Z, your study design is terrible, maybe then you'd go back to the lab or you go back to where you're thinking about things and you redesign the experiments. But most things don't suffer from being fundamentally flawed. They need another round of experiments, or they need another set of data, or they need it explained differently, or somebody was crabby that day, or the editor-in-chief hates otitis media and is never going to publish anything in the International Journal of Otolaryngology on otitis media. I mean, I'm using hyperbole, but I mean, I think there definitely are ways to go about really good work. In my career, my lab would tell you, I say this all the time, it's my favorite kind of research. It doesn't matter what the research says, we're going to publish it, right? Like, so it either proves our hypothesis or disproves our hypothesis, or it takes us in another direction. But it's my favorite kind of research where I think we can publish this. And I start lots of lab meetings that way because I think, intellectually curious and scientifically honest data deserves and can be published. So, all right, ending with a sermon, but really ending with acknowledgement of, I mean, so thank you for inviting me to do this. This is a joy to be at this stage of the career, to weave a story like this together. I've had one or two original ideas, maybe a couple more, but the reality of this is that it's been the people around me who have enabled me to do such amazing work. And my pre and postdoctoral students, I would need multiple slides to actually list all of them, but oftentimes they challenged and brought really great ideas. Lots of clinical partners who have helped me, and I've listed some names here on the list of really the most important collaborators that I've had over time. So I'm going to stop there. I don't know, Reggie, I finished. I think you said you wanted me to take 50 minutes or so. I started a couple minutes late, so it's 8:52. I don't know if you want questions or if you want me to just be quiet finally.
No, that was perfect. Thank you for being so generous with your work, with your ideas, and your nuggets of wisdom. That's really well appreciated, especially for our junior investigators. I do want to say to this audience that ASPO agreed to post our recorded webinars, including this one, in their social media. So I think that will just carry some of the ideas here farther than our current network. So I'm really thankful to them, and you plugging them, I think, is perfect.
I didn't know that. I didn't know that before the talk. So very good.
Yeah. Any questions from the audience, from the attendees? We do have representation from multiple continents, so I'm really happy to see that today.
I have a question that was really great, Joe. This is Sarah Clark from Colorado.
Yeah.
Um, really exciting with the getting these samples from the kids and being able to culture the primary cells. I think that's such a great way to get really close to what's happening in those kids. I was curious, you mentioned the cochlear implant controls. Did you also look ever in adults who are experiencing these infections and see differences? I know they're less common, but I'm curious if you looked at that.
Simple answer is no. I'm a pediatric otolaryngologist, and Children's Wisconsin didn't allow me to see patients generally who were over the age of 18, 21 sometimes. But I actually, true story, I did have a lot of patients in my practice who were older chronic ear patients, but most of them had some other developmental thing, Down syndrome and stuff that wouldn't have made them candidates for a research study. So no, but Sarah, it's a fantastic idea. I mean, again, one of the things that really has been fun and is comparing different kinds of avenues. We've published a couple of papers actually, even looking at uh, uh, boys versus girls, and we know that boys have more difficulties with otitis media. We actually have a hypothesis in the lab that estrogen may be protective, and we've done RNA-seq studies that have shown that things in the estrogen line actually may be related, and there's some work to be done there. Again, one of the hard things in getting to this part of the story is that I have lots more questions in other ways, and that's why I hope this does inspire other folks. But I think, Sarah, it would be very easy to do. I mean, it would be very easy to do for somebody that has an adult otology practice to say go in and harvest some samples from adults and just do some comparative analyses. It would be my favorite kind of research, Sarah. It wouldn't matter if it was the same or different, but it would be definitely publishable.
Yeah, perfect. We always love that. Thanks.
Anyone else with a question? Thanks for that, Sarah.
Joe, not so much a question other than a comment. First of all, I loved all your nuggets, even me at my stage. These are things to think about all the time, including publishing. But I just wanted to really thank you, Joe. I mean, your work was really not only pioneering, but really instrumental in my career as well, particularly the biofilm work, as you eloquently described in your talk. And then sequencing the chinchilla genome, amazing. I mean, that really set us up to do some work that really I feel like has moved the field forward as well. We couldn't have done those things in those studies. You have been a great mentor to me, my lab, and our whole group for many years. So I was really appreciative of your talk, but also listening to you today. So I just wanted to publicly thank you for that. And just let you know how instrumental that's been to us in our career. So thanks, Joe.
Well, thanks, Kevin. That's kind of you. And as you know, Lauren is on this slide and was a great mentor to both of us and has been a great collaborator. And so I would just reflect back to you that every mentor-mentee relationship is bidirectional. And if it isn't, you should get out of it, whether you're the mentor or the mentee. And so I've learned as much from you in your lab, I'm sure more than you've learned from me. But just really quickly, since you mentioned the chinchilla genome, this is also just one quick story about, like, literally that idea was hatched in a hotel room in Chicago. So, and talking to people and getting some of these stories is fun because it just shows you how much fun science can be. But we were struggling with this problem forever, and Lauren and Garth, who are interestingly in alphabetical order, the people listed here are in alphabetical order, but they were also two of the most important mentors for me. We just decided, look, we're going to go to Chicago. We're going to lock ourselves in a hotel room until we figure it out. And we literally did that. Lauren came from Columbus, Garth came from Pittsburgh, and I drove down from Milwaukee. And we went to it's I think it's Rosemont, or it has the word Rose in it. The hotel. It's near downtown Chicago, cheap enough, and we literally each got a hotel room for the weekend, and we just I can't remember whose hotel room we sat in, but we literally just started throwing stuff on paper that entire weekend. By Sunday afternoon, we had a plan, and I guess creativity and commitment and being willing to be with your colleagues. None of us cared about the credit of who got credit for it. We all invested money. MCW probably invested a little more because we had to put it up on the genome side. But yeah, I mean, anyway, thank you for allowing me to say another story. I should probably hang up though, because I know there's another much better talk, I'm sure, coming from Dr. Tucker next. But please do send me an email. Reggie, you can send my email out to folks if there's anything else I can answer. I wish I could stay to hear the second talk, and it's rude of me to not do that, and I apologize, but I do have to run, and it's been my pleasure to be with you.
Appreciate everything, Joe. Thank you. All right, that was great. Now we will have Professor Abigail Tucker from the Development and Evolution in the Center for Craniofacial and Regenerative Biology at King's College London. She has an interest in the middle ear from both a clinical perspective and an understanding of how the mammalian ear evolved. And I'm really thankful to Abby for giving this talk. I'm excited. I know most of what she will present today are new, so I hope it really inspires our junior investigators to think about new ideas from her talk. Thank you.
Thanks very much. It's a real pleasure to be here, and really enjoyed that last talk. So that was great. So I'm a developmental biologist, and a lot of the work we do is focusing on the ear. But I'm going to tell you a little bit about what we've been doing focused on ear repair. But before we get to repair, I thought we'd start with the whole sort of life cycle of the eardrum. So a little bit of development and homeostasis before we get into repair. And I'm also going to be talking about different animal models. A lot of the time we're working on mice, and I'm going to sort of show you why we've picked mice. We've just heard about chinchillas. So as we go through, we'll have little sort of pictures that will tell you which animal we're looking at. So to start with, we've already had a bit of an introduction to the eardrum, but it really does have this quite unusual anatomy. Because it's the only place in the body where all the different germ layers of the embryo meet. We've got an endoderm on the inside, we've got a mesoderm in the middle, along with neural crest cells, and then we've got an ectoderm on the outside. And it's extremely thin. So it obviously has to be thin for its function, but that in itself is quite difficult to make a structure that's robust and yet can conduct sound. Oops. Next one. And one of the things that we're sort of acutely aware of is that actually eardrums perforate quite a lot of the time. Here's a couple, well, top one shows a perforation, the bottom is some scarring after healing. And actually, eardrums heal really well. So it's only about 6% of eardrums that don't manage to heal. Those are the ones that turn into chronic problems. So when things heal quite well, we usually associate them with having some sort of innate system of stem cells to give them the regenerative potential. So that's one of the things that we've been looking at, and then looking at what happens in those 6% where we don't get good healing. So we're going to start with the question about how do you make an eardrum in the first place, then look at homeostasis, and then repair. And I've got this rather nice dissection of a mouse's eardrum. because I think it sort of highlights two things, which is that actually when we're thinking about an eardrum, this is the malleus, so one of the middle ear ossicles, and this is its manubrium that inserts into the eardrum to be able to pick up vibrations. And the eardrum is divided into two parts. So in fact, you have a large round like structure that is your pars tensor, which is the bit involved in hearing, and it's very taut, and it's held by this circular ring called the tympanic ring. So we've got this bone that holds the eardrum really taut. And then we've got the second part of the eardrum, which is over the back. Hopefully you can see. Let me just change my... okay, you can see it better now. So we've got this other side over here, which is the pars flaccida, which is much less tense, as the name implies, and it's where if you have pressure changes, that part of the eardrum can sort of accommodate those changes in pressure to stop the eardrum from bursting. And when you look across animals, I think it's really important to sort of understand that everybody's ear is not the same. That if we're looking here in a rather great picture from 1832, drawn by Shrapnel, which is showing the relative sizes of the pars tensor and the pars flaccida. And so, for example, if you're a sheep, your pars flaccida is really large, pretty much the same size as your pars tensor. If you're a cat, it's absolutely tiny. So all these things, when we're looking at eardrums, we can appreciate that actually there's quite a lot of variation across different mammals. So if we jump into the human developing eardrum, you've got to be able to create this structure that has this endoderm on the inside, ectoderm on the outside, and that happens because in the embryo, those two tissues sort of head towards each other, come past each other, and make this eardrum structure. And that's what we're looking at here in a human embryo, where in pink we're looking at on the inside the endoderm. So here's our endoderm, and that's moved in up from the pharynx. And then from the outside, we've got the ectoderm that moves in, forms this very sort of thin plate of cells. And this is going to be our eardrum. So where we've got the asterisk. So you can imagine this structure is going to be the middle layers, and then we've got an inner layer and an outer layer. So that's how we've created the eardrum in the embryo. When we look at this, these tissues have to actually sort of know that they're coming together, and you've got to have some coordination so that they're not sort of missing each other and actually they're heading in the right direction. And one of the things that we've been looking at using both mouse models where we can manipulate things, but also then looking at human embryos to be able to say, is this relevant to humans? Is this sort of coordinated development, and what signals are involved? And we can also look at rather weird and wonderful animals. I've thrown this platypus picture in because this is the ear canal that is forming, and the end of the ear canal is going to form the outer layer of the eardrum. And it's coming over here to meet up with this structure coming up from the pharynx, which is the endoderm. It's got a long way to go. It started all the way up here and was a sort of little finger of epithelium that's migrated all this way. So these cells need to know where they're going. And if you do a reconstruction of the tissues and you sort of try and look at what's heading for what, we get this rather nice image. This one's from a mouse, where the ear canal epithelium I've got in red, and then I've got the tympanic ring, which is that circle that holds the eardrum wide open, in green. And we can see it's like a hand fitting into a glove. The two are sort of coming towards each other and have a really close fit. And if we muck around with some signals, so we can stop signals that are normally produced from the tympanic ring using transgenic mouse models. The result is something like this. So this is an example where we've stopped the incoming ectoderm of the ear canal from being able to respond to fibroblast growth factor signaling. And as a result, the ectoderm doesn't migrate in and it doesn't manage to move towards the tympanic ring, and we don't form an ear canal and we don't form an eardrum. So we've got to coordinate these signals in the embryo. One of the nice things we can do in the lab is culture little pieces of tissue. This is a cultured ear canal. And we thought, well, if we think that the signal that the ear canal is heading towards a fibroblast growth factor signal, if we provide an extra signal somewhere else, can we divert the course of the ear canal? And that's what we've got here. So we've got beads soaked in control medium. The ear canal moves along as normal. If we put in beads that have an FGF source, suddenly we get the ear canal starts to branch and head off in the wrong direction towards one of the beads. So we know that the signals, therefore, are that are controlling this migration of the ear canal and creating the eardrum are controlled by FGFs. Then when you get your eardrum to the right place, on the outside, it's been created by this solid group of cells that have to open up. So we need an open ear canal. We can't just have a sort of closed tube. And so this is, again, looking at a human embryo, and we can see here, this is the a split that is occurring down that ectoderm. So to separate the eardrum away from the rest of the ectoderm. And it makes an absolutely beautiful image. And one of the things we can see here, so if we looked at an earlier stage, these tissues would have been attached to these tissues. So this is this new opening that's being made to make an ear canal on one side and an eardrum on the other. And we can see how thin these layers are. So the ectoderm here is shown, it's got in two layers in green and red, and it's super thin in the eardrum. So something that is, cells lining the ear canal much thicker. And we've looked at how you actually create that sort of asymmetrical divide in order to make such a thin structure. It really is literally two cells thick. This, again, is all in the human, and was carried out by one of my clinical colleagues, Mona. And this asymmetrical splitting only happens at the ear canal. So if you look further back through the epithelium, it's symmetrical. And we can bring that down to actually what's the mechanism. We can show that there's changes in proliferation, and those differential proliferation means that the eardrum splits and starts opening. These cells are showing where the opening is about to happen to create that really thin eardrum membrane. Okay, so to sort of move on from development, I've just shown you that we think FGF signaling is really important for actually causing the ectoderm to head towards the tympanic ring where it's going to insert. And we've got this issue of having to have a solid structure that needs to open asymmetrically to create this thin eardrum. So now I'm going to turn to the work we've been recently doing looking at eardrum repair and taking some of the signals that we know are involved in development, re-looking at them in a repair context. And for this, we've been using the mouse as a model. And the reason we've been using the mouse is because of the really fantastic molecular manipulations you can do. You can take a gene and say, I would like to turn this gene off, or I'd like to turn this gene on, and you can do it in a specific population of cells at a specific time point. So it's extremely accurate, and so I think those things make the mouse a fantastic model. The flip side of that is that mice have, because they're little, they have very little eardrums. And I think it really is just down to that size difference, but they heal extremely well. So whereas a chinchilla, we can say, yes, a chinchilla eardrum really in middle ear is really very similar to a human. One of the things that a mouse can do is it just heals super, super well. It's very difficult to look at what happens with eardrums that don't heal when the mouse just keeps on healing whatever you do. So if we start with homeostasis, so we've got our eardrum now, it's all been set up. So we've got this structure here. But when you have, if you're thinking about your skin, and your skin is continually turning over, and the skin on the top just sloughs off, and that's fine because you've lost the skin. That's very difficult to do when you're at the bottom of an ear canal. So it's a bit like a sort of blind alley. And so these cells have got to go somewhere. And that's solved by having this really interesting directed migration of cells, so that the cells move off out of the ear canal and we lose them out the ear, rather than all the cells compacting and getting stuck. And we can see that from in the human, people have done quite a lot of studies where they've done these little ink dots, and you can follow over time how those ink patterns move and sort of start moving out of the eardrum. And in the mouse, people have used lots of really amazing tools. This is a very nice paper from Aaron Thward's lab, genetically labeling little populations of cells and seeing where they form, where they move to in the mouse. The other thing that's going on to do with that sort of turnover is that there are stem cell populations in the eardrum, and they're not all over the place. They are sitting in defined little niches. So if we're looking at a sort of schematic of the middle ear, two places to do with the eardrum that we really see these stem cell populations is one right in the middle of the eardrum, where you've got the manubrium of the malleus joining, and right at the edges of the eardrum. So we've been identifying those cells. This was from a paper almost 10 years ago, looking at identifying them because they were label retaining, which is one way of identifying a stem cell population. We've recently discovered that those label retaining stem cells also express a gene called axin-2. Axin-2 is a part of the Wnt signaling pathway. And if we just look at where those axin-2 cells here are, and this is all the work of a great PhD student, Olivia, we can see that those cells are found just where the label retaining cells were found, around the edges of the eardrum, along the where it meets the tympanic ring, and in the middle by the manubrium. And these are just showing two different mouse models with a fluorescent version and a blue dye version of showing the same thing. So we've got these axin-2 cells, and because we've now got a label for these potential stem cells, we can follow them and we can say, what are those cells doing? This is just showing that those cells overlap with the label retaining cells, and also from single cell RNA-seq data, we can show that they express many of the markers of quiescent cells. So these fit the bill for cells that are sitting around, not doing very much in the general day-to-day, but are sort of primed so that they're ready whenever there's a repair is needed. One of the rather, I've just thrown this in because it's rather pretty, but this is an example of one of the transgenic tools we have, where we can follow cell populations and clonal arrangements of cells just by looking in these transgenic mice at different times. This is called the confetti mouse. And you can see that why it's called the confetti mouse. It's absolutely beautiful. There are lots of different colors, and each clone will be a different color. And the other thing that's rather important to do is if you then want to look at repair, you have to have a really robust model for being able to look at the repair process. And for that, we use this tiny little otoscope so that we can look inside a mouse's ear and see what's happening. And in particular, we can go in and make sure that when we're making a hole, we're making a perforation, that our perforation is in the same place, it's the same size, or we can change the position and the size, but that we can track how big and where the hole was at the start of the experiment. And so these are some eardrums just showing the general sort of timing of healing for a mouse. So here is a little hole 1 day after a perforation. These have been dissected out of the animal so we can see them. And then at 10 days, we can see that the hole has repaired. There's a little bit of thickening, and it takes usually about 30 days for the eardrum to completely repair. And we can see that process here over a series of developmental time points going up to that 30 days, where we have an unperforated ear, so we can see how thin the eardrum is. Here now a perforation has been made in between the manubrium and this tympanic ring side. 5 days, we can see this massive thickening of epithelium. So the epithelium definitely seems to lead the way in the repair process. It's the first tissue to respond. And then by about 10 days, we've now bridged the gap. We've got a bridging of the gap, and we start forming a sort of scab of that. So a lot of that epithelial tissue that was used for creating the bridge then falls off as a scab on the outside. And then by 15 days, it's almost as thin as the unperforated ear. And by 30 days, it really is as thin. So it's a very good system for healing. So we can then say, during this process, using our transgenic mice, we can follow what happens with different cell populations. This is an example where we've looked at neural crest cells. And so all cells that are neural crest derived will be red. And we can say, where are those cells? How are they contributing to repair? At what time point do they come in? And this is the bit I just want to sort of home in on. At 5 days, we've got our hole in the middle, and we've still got not very many neural crest cells around. But by 10 days and 15 days, we've got a lot of neural crest cells. So the neural crest plays a role in the repair, but it comes in much later. So it's really not moving in until about 10 days. We can see that also we've got changes in the vasculature. So this is another mouse model that we can use to follow the vasculature. So again, this is an unperforated ear. Perforation now here, and we can see the vasculature moving in around this region, and then it starts to retract and move out of the eardrum again after the repair has finished. This is now looking in section, but again with that idea of which layer, what layers are doing what in this process. And we can see we've got a hole at 5 days. 7 days, we've now got a bridge. And that bridge is being made by this epithelium, which is in green. And there's still the red is our middle layer. That middle layer hasn't moved across yet, but by 10 days, that middle layer has now moved across. So we can chart the timing of how the repair process occurs. And then going back to those potential stem cells, we can look at our axin 2 population and say, what do those cells do? And actually, very interestingly, they already start moving in at 5 days after perforation, but absolutely loads of them at 10 days. And then those cells continue to recreate the new repaired membrane when we look at later stages. So not only are those cells sort of label retaining and sitting around the outside of the eardrum and near the manubrium, as soon as there's a perforation, within 3 to 5 days, those cells have already started moving in to bridge the gap. So we think they're a really key population. And again, the kind of thing we can do in mice, we can say, okay, if we think they're important, what happens if we mess around with Wnt signaling and stop those cells from expressing and being able to be activated with Wnt signaling? And the result is, here's our little hole down here. This is our control up at the top. So all those red cells have moved in. Here we've got a hole still. And actually, it's caused by the mesenchyme doesn't bridge the gap, and as a result, the ectodermal cells get in the way and stop healing from happening. So actually, this is quite a good model of why something might not heal well, is if the epithelial cells got in the way and there was a problem with Wnt signaling. So I'll end with looking at otitis media, because that is one of the things that we're very interested in the lab. We've been looking mainly at syndromic models, mouse models, for example, of Down syndrome, DiGeorge syndrome, branchiootorenal syndrome, all syndromes that have really high incidence of otitis media, and the mice also have very high levels of otitis media. We're also now moving into looking at more microbiome-influenced otitis media, the sort of more acute rather than this sort of chronic that we see in these cases. But I just want to show you this one because this is our DiGeorge syndrome mouse model. We can see that here the mouse has got absolutely, its middle ear is full of lots of inflammatory cells, got thickening of the epithelium. It's really not a very happy middle ear. And when we perforate that ear, we can see that it really doesn't heal very well. So the healing process is influenced by otitis media in the middle ear. So in the control, this is the scab on the outside that is about to fall off. And then this is our reformed eardrum membrane. And over here, the tissues are all completely unorganized. We can see the infiltrated cells that have all sort of become part of this scab formation. So this is something that we think is a really interesting avenue to further explore. So that's my model, therefore, and so we think that we've got these stem cell populations that are sitting right in the middle of the eardrum, but also at both sides, around the tympanic ring. And in a repair situation, it's the epithelium, so that on the outer layer, retracts and then proliferates hugely to bridge the gap. And once the epithelium has bridged the gap, then we've got our other layers, our endoderm and our mesenchyme in the middle. That's when that can reach over and reform all the layers, at which point we get a scab falling off on the outside and then remodeling. But these axin 2 cells playing a really essential role in reforming the eardrum. So yeah, I've come to the end of my time. So hopefully that showed you a little bit of the things that you can do with mouse models. I really do think they are a very useful system, but as with all these things, you have to have them as a bit of a caveat that they are not the same as humans, but we can, as long as we know that, I think they're a very useful model. And I'll end by saying thank you to my lab and particularly to the funders. We've just got a grant recently from the Royal National Institute for the Deaf in the UK. So that's going to allow us to continue this work and particularly look at that link between otitis media and eardrum perforations. Thank you very much.
Fascinating. I'm really thankful that you gave this talk. I don't think anybody else in the world is doing this kind of work at this level, Abby. So I'm really thankful. Any questions? Yeah, it's so nice. Any questions from the group?
I have a quick one.
Go ahead.
Yeah, so wonderful to see all the like fluorescence images and histology kind of showing the process. Can you comment a little bit on the different stains or dyes you've tried? I know you mentioned some in your talk, but
yeah, so we use a lot of mice that have had fluorescent genes incorporated into them. So, for example, for the vasculature, we were using a mouse that has a jellyfish gene that's been inserted into its genome that can be turned on specifically in those cells in the vasculature, and so that we can follow them. When we give the mouse an injection, suddenly goes lights up. At the same time, we're also using a lot of immunohistochemistry to look at, so for the ectoderm layer, you can use E-cadherin, for example, or one of the keratins to be able to look at that layer. So we sort of mix both approaches. And I've just actually started on some spatial transcriptomics of the mouse ear, which is where you can look at 5,000 genes in one snapshot of one piece of tissue. And I think that's going to keep us going with more genes and more markers to be able to look at for quite a few years. Thanks.
yeah, wonderful. Thank you.
Anyone else with a question?
Well, if not, I'll stop sharing anyway.
Maybe I have one. And thank you, Abigail, for the great talk. I hope that you can hear me.
Yes.
Well, I followed a little bit your work. I'm not an expert on the topic, but I was wondering, this more stem population is also found in human, and is there a prevalent location where this population is found?
So we've looked, so we can't do these sort of label retaining cells in human samples, but we can look at axin 2. So we've had a look at that in our late developing human fetuses that we have access to. Actually, I should have said many thanks to the Human Developmental Biology resource, which we have in the UK, because, I mean, we wouldn't be able to do this work with human embryos and fetuses without them. But the axin 2 has almost identical expression in the mouse as the mouse, and found at the border around the tympanic ring and at the manubrium in the middle. And in humans, there is some evidence to suggest that depending on where the perforation is, that might be a reason why it doesn't heal. And I think certainly if you disrupt those stem cell populations, you're really going to affect the healing process.
Well, aren't they competing with each other? So you have the epithelium migrating into the perforation, and also this population of stem cells trying to proliferate. So do you see this competition in the healing?
So we don't see it so much in healing, and that might be because in the mouse it happens so quickly. So you suddenly get this sort of burst of proliferation and everything moves in. So the normal migration out stops. I think in humans it'd be really interesting to sort of have a look at what happens with the migration. And some people have done those ear ink spot experiments and looking at perforated ears and non-perforated ears. So I think there's a possibility to sort of look at that in humans as well.
Okay, and the final question, and sorry to take so many questions. You showed that there is this thickening of the when there is the healing, but is this thickening majorly cellular, or you have a lot of ACM, a lot of collagen being secreted in there that induces this thickening?
It's mostly a proliferation. So we see very high levels of proliferation, particularly in the ectoderm. So it's the outer layer. So it's the outer layer that suddenly proliferates. I mean, it goes to almost, I would say, like 30 times the normal thickness of the eardrum just in a few days. So that's very cellular. The underlying mesenchyme is much slower. That doesn't proliferate to the same extent. So we think that the ectoderm is the really important thing for making the bridge and closing the hole. And then once it's there, it's like a scaffold, and the other tissues can move over it.
Okay, thank you very much.
In the interest of time, we have to stop here. Thanks again to Abby and to Joe. If you have questions, we can forward their emails to you. You can just email me or Landon. And I might have my own question, Abby. I'll probably email you.
Yes, definitely.
Thank you. I just have two announcements. So for the year two mentees, because of some updates with RedCap, we will send the questions for the quiz, which also is kind of like your attendance by Monday instead of today. Sonia just wanted me to let you know. Oh, Sonia's still here. Sonia, if you want to say something, you can unmute too.
Yeah, I mean, you basically said it. So we'll send out the survey Monday because RedCap is doing an upgrade in Colorado, and they start at 9 a.m. this morning. So I'm going to send you guys the survey on Monday morning.
All right, thank you. And I hope that we can see you in our next webinar. The next one will be in January. We will have chronic suppurative otitis media as a topic for scientific and for non-scientific difficult work environments. We have a nice panel with Mudbuta from the UK and Peter Santamaria from Pittsburgh. And then Rita Lee will have our other webinar. So see you then. Thank you. Bye.
Thanks very much, everybody.